The third part of this lecture is devoted to other three catalytic strategies used by the enzyme, which are preferential binding to the transition state complex, proximity and orientation effects, metal-ion catalysis. Just looking at this energy diagram for uncatalyzed and catalyzed reaction, one can suggest that in order to bring the energy of transition state down and by doing this facilitate rate of chemical reaction, the transition state should be tightly bound to the enzyme. And this is realized in nature, and this effect is called preferential binding to the transition state complex. Just looking at the induced fit model that states that enzyme doesn't have site optimal for substrate binding, one can again guess that enzyme has a site optimal for the binding of transition state. For the next several slides, we'll illustrate you several examples of transition state analogs. I have to say that transition state analogs are considered good inhibitors of the enzymes, and as we will be studying, inhibitors of the enzymes can be used as drugs for treatment of human diseases, for example. One transition state analog that I would like to mention is shown here. It's diisopropyl phosphorofluoridate, or DIPF, and it can specifically bind to active sites of serine proteases, followed by nucleophilic substitution of fluorine with serine to form covalent complex, which is stable enough under physiological conditions, resulting to irreversible inhibition of serine proteases. Let's take a look at the mechanism of action of this inhibitor. This picture brings you back to the tetrahedral intermediate found during catalytic action of serine proteases. So this is tetrahedral intermediate. And on the next slide, I have redrawn it next to DIFP. So this is the tetrahedral intermediate. And this is DIFP in the active site of serine proteases. You can find many similarities of this compound with the tetrahedral intermediate. First of all, both phosphorus and carbon are tetrahedrally coordinated, which means that DIFP has geometrical complementarity to the active site of serine protease. Second thing that I would like to draw your attention to is the negative charge on this oxygen that is located in oxyanion hole, and the delta negative charge on this oxygen that is also located in oxyanion hole. More similarities. Nitrogen can form hydrogen bond with hydrogen provided by the imidazole ring, and this oxygen also is capable of forming hydrogen bond. As you can see, in this complex, phosphorus is close to serine, to the active serine residue for nucleophilic attack, and that's what happens. Fluorine is a good leaving group, and oxygen forms a covalent bond by nucleophilic substitution of fluorine in DIFP. Second example of transition state analogs that I would like to cover in this course are shown on this slide. They are inhibitors of proline racemase, which is an enzyme that catalyzes conversion of L-proline to D-proline and vice versa. So eventually a racemic mixture in which 50% L-proline and 50% D-proline is present. And the reaction is passing through a planar transition state. The important feature of this transition state is planar conformation of alpha carbon right here. Indeed, at the first stage, proton is abstracted from alpha carbon and carbanion with lone electron pair perpendicular to the plane of the molecule is created, and then it is in aqueous environment, in the presence of protons provided by water, it is protonated to form either diproline or L-proline. If you take a look at the structure of transition state analogs, you will see that alpha carbon in both cases is in sp2 hybridization state, thus providing planar conformation of this portion of the molecule. And this structure resembles the planar transition state structure. It was shown that the two transition state analogs shown here bind proline racemase about 160 times tighter than either L-proline or D-proline. And the third example of transition state analogs that I would like to talk about is those that inhibit adenosine deaminase. This is an enzyme that catalyzes deamination of adenosine. So you can see adenosine, and it is converted to inosine. I have to say that this is a biologically important reaction because we know that inosine is found in the structure of tRNA and actually many other RNAs. And the reaction involves addition of water molecule to the double bond, CN double bond, and formation of a transition state intermediate, which is characterized by non-planar conformation at carbon number 6 in adenosine, in former adenosine. So this atom is sp3 hybridized, and the conformation is quite different from planar conformation of both adenosine and inosine. And then the amino group and OH group are not stable at one carbon. Amino group is released as NH3, as ammonia, and inosine is formed. So a good transition state analog shown here would have all the same purine structure, and the main modification here is sp3 hybridized carbon number 6, and this compound is stable because CC bond, this is a CH3 group, CC bond makes this structure stable. Next catalytic strategy that I would like to cover is proximity and orientation effect. Proximity and orientation effects increase rates of catalyzed reaction in all cases. For all enzymes, we have this effect. And they achieve increasing rates because of the three major factors. First of all, they bring substrates in close proximity to each other. Second, they bind substrates in proper orientation towards each other. And third, they freeze transitional and rotational motions of both substrates. One example is again taken from serine proteases, is water molecule positioned next to the ester bond that is about to be hydrolyzed. You can see here first that the two substrates are in close proximity, and this is achieved by binding water molecule in exact position. Now oxygen is oriented towards the nucleophilic carbon, and again this is achieved by orientation of water molecule and perhaps by formation of this hydrogen bond between imidazole ring of histidine residue and water. And the transitional and rotational motions of water freezed, again, it's achieved because of binding water to histidine residue, and also it is achieved by covalent binding of this second substrate to a specific position within the active site of serine protease. This strategy, proximity and orientation effects can increase rate of reaction in comparison with uncatalyzed reaction by up to 10 to the 7 times. So we covered all these strategies, and the last strategy in our list is metal ion catalysis. So let's talk about transition metal ion catalysis. So metal ions that participate in catalysis are listed in this on this slide, and they participate in 3 different kinds of reactions or effects. First, they can bind substrate and orient them towards reaction. They can mediate redox reactions, for example, iron or copper that have variable oxidation states. Then they can participate in the redox reactions. And second thing is electrostatic stabilization of negative charges. So you can say it's electrophilic catalysis. And one example of such electrophilic catalysis can be illustrated using carbonic anhydrase as an example. Carbonic anhydrase is an important, physiologically important enzyme found, for example, in human blood, and it facilitates hydration of carbon dioxide to produce hydrocarbonate ion and H+. Here on this slide you can see 3-dimensional arrangement of carbonic anhydrase, and you can identify an ion right in the center of the structure, and this is zinc ion. Zinc ion is bound by 3 histidine residues, and the 4th valency is occupied by chemically bound water molecule. The water molecule also interacts with histidine 64. You can see that histidine acts here as a general base to remove proton from water molecule. And the catalytic action begins with deprotonation of water, and this is achieved due to polarization of this strong polarization of this OH bond due to electron attraction to the positively charged zinc ion. So this group becomes quite nucleophilic because of the full negative charge on oxygen, and this facilitates the nucleophilic attack on a central carbon in carbon dioxide. So this is a quick reaction. So you can see now in the active site we have hydrocarbonate ion formed by reaction of OH with carbon dioxide, and then next stage is binding of water which substitutes and releases hydrocarbonate ion in solution. So this is the original state that we started with. I have to mention that carbonic anhydrase was already mentioned in this course when we studied hemoglobin assisted transportation of oxygen and carbon dioxide in blood, specifically carbonic anhydrase hydrates carbon dioxide in blood near the actively respiratory tissue. So metal ions are cofactors of enzymes, and cofactors are in general all molecules of non-peptide nature that helps enzymes catalyze chemical reactions. Cofactors are divided in metal ions and coenzymes. Coenzymes all are organic molecules, non-peptide organic molecules. Coenzymes are further split into two groups, co-substrates and prosthetic groups. Co-substrates are typically loosely bound to the enzymes and often undergo transformations, chemical transformations during the reaction. While prosthetic groups, they are tightly bound to the enzyme. This binding can be covalent or non-covalent, but in all cases, prosthetic groups do not exchange with the environment. They are not released from the enzymes during the reaction. One example of a co-substrate that I would like to mention is nicotine adenine dinucleotide, which participates in redox reactions in many biochemical processes. And NAD plus is an oxidized form of this coenzyme, and NADH is a reduced form. Alcohol dehydrogenase is one of the enzymes that use NAD plus co-substrate. As you can see, NAD plus in its oxidized form takes out 2 electrons from carbon number 1 in alcohol and produces acetaldehyde and reduced form of NADH. So you can see that co-substrate underwent chemical transformation from oxidized to reduced form during this process. As an example of prosthetic group, I can bring to your attention heme. We already studied heme when we talked about myoglobin and hemoglobin. In these two proteins, heme binds oxygen for further transportation oxygen in blood, and heme is also a part of other proteins which are called cytochromes P450, and the structure of P450 is shown on this slide with heme bound in the central position. Cytochromes P450 are responsible for oxidation of broad range of compounds, and they come in many varieties. They found in plants, animals, and specifically in animals, they are responsible for oxidation of cyanobiotics, compounds that are not expected to be found in the bodies, and by oxidizing cyanobiotics, they detoxify organisms of animals.